So for today's talk, I'm going to focus on the importance of rethinking our approach to data visualization and statistics education for small sample size data sets and investigators who work with those data sets, and increasing transparency. So the reproducibility crisis, as I'm sure many of you know, has a lot of moving parts. So people are thinking about and depositing. They are thinking about registering studies. They're thinking about a lot of study designs, power calculation, and statistical analysis. So that where for data visualization? Well, I would argue that data visualization is important because data visualization is really the foundation of our shared scientific knowledge. So many times the raw data underlying a data set or a paper aren't available. And so everything that we know and everything that we're basing our conclusion on is limited by what the authors choose to present in that paper. And that means that the figures are going to be especially important. That's where we often put the data supporting key findings for the paper. And so if authors are making poor decisions about the types of figures that they use, if they're using figures that aren't transparent, that than providing the important information that we need to critically evaluate that data, then it means for to use that data and get the most out of the data set in the future. So today I'm going to focus on several different topics. I'll start off with a quick overview of the problems with our current practices for how we present data in small studies, and that includes the inappropriate use of bar and line graphs to present small sample size continuous data. I'll talk a little bit about why this happens. So how did we get to a place where the type of figures that basic scientists use as standard practice are things that our statisticians would say really just aren't appropriate at all for the type of data that we use them for. And I'll talk a little bit about some education so that the new generation of scientists comes in with better skills and better understanding. And I'll spend the last half of the talk focusing on solutions. So I'll talk about how you can improve your static graphics to eliminate some of the bar and line graphs and use figures that are more revealing and informative. I'll demonstrate a couple of our free tools for creating interactive graphics for scientific papers. I will show a couple of our interactive graphics resources, but I've also sent out from my Twitter account a few days ago a list of free online resources that you can use to create better graphics for your own scientific publications. So if you have Prism or other expensive software, that's great. You actually don't need it to do anything that I'm going to show you today. There are already tools out there that will allow you to do these things. And I'll close with just a little bit on the importance of meta-research as a tool for addressing some of these issues in rigor and reproducibility. And if there are problems you are concerned about, how you can use meta-research on your own to move your own field towards solutions. So our work, we typically focus on small sample size data sets, so investigators who are working with less than 15 independent observations per group. And in the era of big data, I think it's really important just to spend a minute talking about why these small data sets are so important and why we should be thinking about them. Small sample size data sets are extremely common in basic biomedical and biological sciences, as well as in small sample size human studies and translational science. And these studies should not be overlooked. They're critically important because they influence decisions about what agents go on to clinical trials and to further study and further research. The clinical trials process, as you all know, is time-consuming, it's expensive, it requires a lot of resources and time from investigators, and it places a lot of burdens on patients. And so that means that we want to have as much information as we possibly can when we're choosing which agents are going forward so that we can make the best use of resources possible and make good decisions. And in order to do that, we need to have these small sample size studies presented in a clear and transparent way. The other thing that I think is really important to mention is that in a lot of the NIH writings on reproducibility and rigor, they are focused on the preclinical research, which is by nature small sample size studies. So clinical trials have undergone a lot of changes in the last 20 years regarding how they are reported, designed, and conducted to eliminate or mitigate some of the problems with reproducibility rigor. And those things are just starting to happen in the preclinical sphere. So that means preclinical research still has a lot of problems, and there's a lot of things that we can do to improve reproducibility and rigor in these small sample size studies. So from the NIH perspective, looking at the preclinical research is definitely somewhere we should be focusing whenever we're thinking about reproducibility and rigor. Okay, most of us never get visualization training that we need in order to do our scientific jobs. So our visualization training is often we get our first data set, we turn to the grad student at the desk sitting next to us and we say, what do I do with this? And they say, you make a bar graph, you make a line graph, and we continue doing that for the rest of our careers. This isn't a very good idea, and it's not a very good way to train people in visualization. So today I'm going to give you a little bit more information on how we can design better figures and why that's important. So an effective figure needs to do three things. The first thing is that it should immediately convey information about your study design. So I should, within seconds of looking at your figure, I should know whether you're comparing a set of independent groups, whether you have longitudinal or repeated measures data, or whether your data set includes clusters of non-independent data, so things like replicates or animals from the same litter. Figure should give me all of that information. The next thing that the figure needs to do is illustrate the important findings for your data set. So again, within a few seconds of looking at the figure, I should be able to get a sense of what it is that you're comparing or assessing and what your key findings are going to be. And then the third thing is really critically important, and that is that your figure needs to allow your reader to critically evaluate your data. This is what differentiates science from marketing, right? So in science, we need to be able to look at each other's data, to critically evaluate it, to draw conclusions, and then make decisions about how we can best move the study forward and advance knowledge by conducting other studies. If the data are presented in a way that don't allow you to critically evaluate the data, none of that process happens. And that's a huge limitation to our scientific method. So this is the major limitation of the types of figures that we use now, the bar and line graphs, that they don't allow the reader to critically evaluate the data. And just so we're clear, as Dr. Haywood mentioned, my main interest is in preeclampsia research. And my interest in improving data presentation actually comes directly out of my work in preeclampsia. So with preeclampsia, it's a syndrome, and we know that while women all end up with the same symptoms, they tend to get there in different ways. So some women have a lot more maternal pathways that are contributing. Other women have a lot more pathways related to the fetus and placenta. And the contribution of those maternal pathways and the fetal and placental pathway differ from woman to woman. So that means that for any marker that we're looking at, we expect that it's going to be abnormal in some women who get preeclampsia, but perfectly normal in other women who get preeclampsia. And our goal right now as a field is to understand how we can identify which different subgroups and which women have normalities in which pathways and which women have normalities in other pathways that are also relevant. So if we want to understand heterogeneity, then we need to start presenting our data differently. A bar graph is what we use if we want to mask heterogeneity, right? There are no subgroups in a bar graph. So we started off 2 years ago with our first meta-research paper, which was published in PLOS Biology, and it was titled Beyond Bar and Line Graphs, Time for a New Data Presentation Paradigm. And this paper went viral online on Twitter and Facebook among the scientific community within about 48 hours of publication. And it was picked up by Nature News and the Twitter response at that time as well. And it was viewed more than 100,000 times in the first month that it was published. Since the time that it was published, it has had a number of effects. So it has led to policy changes in several different journals, including PLOS Biology, eLife, and the Journal of Biological Chemistry, that are now discouraging or banning authors from using bar graphs for continuous data, at least for small sample size data sets. Nature also came out with a policy change a few months ago that applies to all of their affiliated journals as well. We've also know anecdotally that a number of editors and reviewers have been using the paper to request better data visualization for papers that are going into different journals. And it was also instrumental in initiating the Bar Plots Kickstarter campaign, which targeted neuroscience journals and general circulation journals to improve data visualization policy and get rid of some of the bar graphs. So the paper had three different parts. We had a systematic review that looked at quantifying how do we present our data now and what are the data presentation practices. We had a clear explanation of why our current data presentation practices are problematic. And then we also had templates that investigators could use to make dot plots and better graphics that were done in Excel. So we looked at 700 papers that were published in the top 25% of physiology journals over a 3-month period. And we found that almost all papers were using bar and line graphs to present continuous data. So 85% of papers had at least one bar graph of a continuous data variable. 61% had a line graph. When we looked at papers that were using figures that showed the data distribution, those papers were rare. So only 15% of papers had a dot plot, and 5 to 8% had a histogram or a box plot. So when it comes to figures that showed a data distribution in the basic sciences, we tend not to use those, and that's a problem. We also found that the sample sizes were very small, and in most cases less than 10 per group. So when we looked at the minimum sample size for any group that was shown in a figure, in 75% of papers that sample size was 6 or less. When we looked at the maximum sample size for any group shown in a figure, in 75% of papers that was 15 independent observations or less. So these are very small data sets. We also found that investigators who are using bar graphs are predominantly using them to show the standard error, which tells you about the accuracy of the mean and not the variability in the data. So 78% of papers with bar graphs were using those graphs to show the standard error. Only about 16% were showing the standard deviation. And finally, we found that these data presentation practices were so ingrained that even amongst papers that use some form of non-parametric analyses, more than half of authors still chose to present data that were analyzed non-parametrically as mean and standard error or mean and standard deviation. And those summary statistics aren't appropriate when you're using a non-parametric test. So we use a lot of bar graphs. Who cares, really, and why is that a problem? Well, one of the reasons it's a problem is many different data distributions can give you exactly the same bar graph. And your actual conclusions may be different based on the actual data set. So here we have a bar graph, and then on the right we have 4 different dot plots of data sets that will give you exactly that same bar graph. And there are p-values for several different types of t-tests below. So t-tests for equal and unequal variances, as well as a non-parametric Wilcoxon test. So if we look at our first data set, we seem to have slightly higher values in our second group compared to our first group. And this is a small difference, but it might be one that we're interested in pursuing. When we look at our second graph, the higher values in the second group really seem to be driven by this one outlier. And that's something that we might be much less likely to follow up on. When we look at our third group, there's some suggestion that the data distribution might be bimodal. So we would want a larger data set in order to confirm whether or not the distribution is in fact bimodal. And we'd also want to know, is there something different about these observations compared to these? So are these the males and these the females? Is there some other variable in our data set that we should be accounting for? And then in our last data of unequal n. So our data points in the second group are clustered toward the top end of our range for the first group. However, we have far fewer data points, so it's entirely possible that we have just underestimated the variability. And if we had more data points in here, we might see much less of a difference between these two groups. So we would probably want more data points in that second group before we went towards any kind of conclusion with that data. So the next question that I always get is, okay, well, let's say I know my data are normally distributed. Can I use a bar graph then? And the answer to that question is still no, for a couple of reasons. First, it still is not going to allow you to critically evaluate that data. So you've said, I know my data are normally distributed, but this is science, and we want to see the evidence of that. We want to see the data showing that so that we can say, yes, we agree with your conclusion, that's correct. And that's how we would have done it too. The second thing is the bar graph actually draws our attention to all of the wrong things in the data. The end of our bar graph is typically going to be just of the highest group, or the highest error bar for the highest group shown in the graph. And when we do that, we actually omit values from the graph that are actually observed in the zone of invisibility. These are these high values here that occur in the data set but don't actually show up in the graph. And then we have a second problematic zone here. Now, when we make a bar graph, we typically start the y-axis at 0. In some cases, 0 is a physiologically or biologically meaningful value. In many cases, it's not. Variables where 0 is simply not possible. It's something that's never going to occur. And so we can end up with this low set of values on our graph that aren't a value that we would ever actually observe in our data set. They have no physiological or biological meaning, and we call this the zone of irrelevance. So when you make a bar graph, what you're drawing attention to is you're missing the zone of invisibility, you're putting a lot of weight on the zone of irrelevance, and you're arbitrarily assigning importance to the bar height instead of focusing your reader's attention on how the difference in means compares to the variability in the data, or how much overlap there is between groups. So just to emphasize, our interpretation depends on what we see. So with a bar graph, all I can really look at is, do these groups look different on average? There's no information here about the sample size that might tell me how confident I can be in these conclusions. There's no information about the data distribution. There's really nothing that allows me to critically evaluate the data. In contrast, and that turns the reader into a passive observer. In contrast, when I have a dot plot here showing the data points, I immediately get a lot more information that causes me to actively think about the data set and about the conclusions that are being offered. So for example, I might say, okay, the thing I see is that this small sample size data set, so I should be cautious about these conclusions. There's a lot of uncertainty here. I might say this group could potentially be bimodal, and I might want more observations to check that. I would see that I have a pretty clear outlier here. I might also see that while this group has high values, it's also the smallest group, and we've probably underestimated variability there. So again, there's a lot of uncertainty into whether this high bar is really meaningful and different from the other groups. So in conclusion, all of the data presentation methods that we use are a reflection of reality. And it's really important to use methods that minimize distortion. Looking at your data in a bar and line graph is kind of like looking at a wavy pond. Maybe you're looking for a duck and you see it and you think that it's a duck. But before you waste a lot of time and effort in following up on those findings and pursuing something, you want to be confident that that thing that you're seeing is actually a duck and not just an oddly shaped potato, right? Nobody wants to waste money looking for an oddly shaped potato when they thought they were chasing down a duck. Okay, so why does this happen? So to give you an idea between the business community and the stats community, I have two statisticians that I work with. And when I went to them and said, you know, this is the problem that I'm worried about. Scientists use bar and line graphs all the time. Here's my data, and I'd like to do something about this. One of them didn't understand what I meant. I said, you know, she said to me, like, how can you use a bar and line graph for continuous data? I don't know what that means. And so I had to explain that the bar is the mean, and then the error bar is usually the standard error, the standard deviation. And she was very confused about why anyone would do this, and it took me 10 to 15 minutes to explain. The other one said, I don't believe you. And I said, but I have data, you can't not believe me. And she said, I understand, like, I see your data, and if that's correct, that is absolutely horrifying. I just can't believe that anyone, much less a field of people, would be making a mistake that's this basic. And I said, well, if you're following standard practice for your field, you're never going to be asked to justify or explain that practice. You're just doing what everyone else is doing, and everyone accepts that that's the way you do it. So we get here. How do we get to a place where the practices that we follow are basic science, as basic scientists, are things that people with more training in statistics and in data visualization would say are completely inappropriate? We in a second paper that we published in PLOS Biology, and one of the things we found was that while statistics are essential for anyone who is reading papers or publishing in the basic sciences, statistics training is not always required for a basic sciences PhD. So we started off with our data set again of 700 papers published in the top 25% of physiology journals. And we found that 97% of them included some statistical analysis, which is probably not surprising to any of you who picked up the journal. Like, all the papers have no one surprised there. What we did was we looked at the top NIH-funded physiology departments, and there were 80 of them. And we looked at whether or not students were required to take a statistics course for the PhD programs that those departments were participating in. And we found that in about 2/3 of the cases that statistics was required for some or all PhD programs that the department participated in. The remaining third, it wasn't required, and there were a mixture of different strategies. So about 10% included stats in their program as a recommended elective, 10% included it as an elective, but didn't specifically recommend or encourage students to take it. And for the remaining 12.5%, statistics was not required, it was not an elective, it simply was not part of the program. And the paper is, among our 80 departments, we had about 5 that had just changed to requiring statistics the year that we did the survey. So if you think about people who did 5 years ago, I suspect these numbers would look substantially worse than they do now. The other thing we found is that even when students are taking to take a statistics course, that course found a lot of physiology students who were taking courses with titles like Statistics for Public Health, which is large sample size data and it's completely different. A lot of departments don't have statistical expertise in health or in-house, so they will go out to epidemiology or biostats or another department, public health, psychology, another department that has more statistical expertise to get stats training for their students. And the result of that is you often have an instructor who is used to working with large sample size data and assumes that their students are going to be doing the same thing, teaching a group of students who work with small sample size data sets with questions and study designs, and neither realizes what's going on. So if you look at the statistical problems that we have in the basic science literature from the perspective of instructors teaching, assuming that students are going to be working with large sample size data sets, students hear it, assuming that it applies to the small sample size with practices accordingly, then the problems that we see make a lot more sense. So we firmly believe that basic scientists really need to be actively involved in designing stats curriculum and in making sure that instructors understand what questions their students are asking, what techniques they're used to seeing in the literature for stats, what the problems are with the way stats are typically done, what sample sizes the students are used to. And both statisticians that I worked with, when for them, and when they started reading basic sciences papers, they said, okay, if I had known that this is what my students were doing, I would have designed my courses completely differently to begin with. And so at Mayo Clinic, that's one of the things that we're doing now. We are developing new curriculum for our basic scientists at Mayo, and once we have that curriculum, it will also be available online for anyone else at any career stage who wants to use it. So we do a number of things that are different when we're designing our courses. The first thing is we focus on data visualization first, second. So teaching data visualization is a lot easier, it's a lot less intimidating for many students, and when students start seeing their data, it naturally leads towards a whole range of statistical questions. It piques their interest and it makes it much easier to move into the statistical topics naturally. The next thing we're doing is targeting misconceptions and misskills that are common in the basic sciences. So misconceptions would be things like you should present continuous data using a bar graph. Or if you find a significant effect in a small sample, it must be a really big effect. Or it doesn't matter if your study is underpowered as long as you find a significant effect. So these types of things are all incorrect, but they're all things that many basic scientists have been taught to believe. And we address those things head on with our students because we know they're gonna go out into a world where these practices are common. So if they haven't heard them and they don't know what the problems are, it's going to be very tough to make any kind of lasting cultural change. The misskills would include things like analysis of clusters of non-independent data, so replicates or mice from the same litter. Basic scientists work with these type of data sets all the time. We're never taught how to analyze them, how to tell whether your cluster design is between group clustered, within group clustered, or between and within group clustered, why that matters, and how it affects, because every one of those things requires different stats. So we teach those skills up front so that we know our students have them or are at least aware of the issue. We also use a visual approach to learning. So we'll create a set of visualizations and formula a lot of math. We'll start with a visualization and use that to have a conversation about the issue and to introduce the topic. And we're also creating simulation tools to allow students to really play with some of the concepts and the rules that they get from their statisticians so that they can understand what considerations and thinking are going into the rules that they finally get, and how arbitrary some of those rules actually are. Because the thing that's been striking for me is how much of what I was taught in basic in my basis statistics training that I have had to relearn as a result of going through this process of critiquing the literature. So here's an example of the type of visualization that we would use in one of our simulators to teach basic concepts. So here we've created a data set that has a mean of this black line, and then plus or minus 1 standard deviation is shown by the gray shaded region. And then we've just drawn random samples from that distribution. And we did it twice, once with an N of 5 per sample, and once with an N of 20 per sample. And we're just looking at how much error there is in the summary statistics and how that depends on your sample size. So right away that you can see that if you have an N of 5 per group, there's a lot more error in your estimated summary statistics. They're much less accurate. So the means are further off in the N of 5 compared to the N of 20. The standard deviations are also further off. And the problem is that so if you do your one experiment with your N of 5, maybe you have this sample here, tiny standard deviation. Or maybe you have this sample all the way out here that's way off, or this sample over here that's also way off, but in a completely different direction. If you're working with a small sample, you're going to have less accuracy in your estimate, and there's no way to know which of the specific samples from this or any other possible sample you might have drawn. So you don't know how far off you're going to be. So this is the type of visualization that we use to show that with small sample sizes, means and standard deviations can be very inaccurate. We then go to something like this, which helps to quantify, okay, accurate. So here's our N of 5, and here's our N of 20, and we're just looking at the error in the sample mean here. So this is no error at all, what it should have been in the population. If we're up here, then the mean is a half a standard deviation off here, or sorry, quarter standard deviation off, half a standard deviation off, 3/4 of standard deviation off, 1 standard deviation off. So the higher you are, the more error you have. And we can look at the N of 5, we can see that small error, so less than a half a standard, or less than a quarter of a standard deviation. For our N of 5 per sample, only 43% of our samples are meeting that criteria. But if we go up to an N of 20, 73% of our samples are meeting that criteria. So this helps students to think about, what do I get? What's the benefit if I go to an N of 15 or an N of 20 or an N of 30? And is that possible for my experiment? This is another example of a simulator that we've created to talk about the issue of normality. So many of us in our intro stats course were given some kind of rule like, if our sample size is smaller than 10 to 30, you should just go straight to the non-parametric test because you can't tell whether your data are normally distributed or not. Whatever you get depends on what you had, and I've heard people give between 10 and 35 for those values. So, okay, where does that number come from? Who made that up, and why was it that number? And if that number is so accurate, every statistician number. The simulator is designed to help questions. So we start off with a visual simulation, and we say, okay, just using your eyes, how your data are distributed. A normal distribution, a skewed distribution, and a bimodal distribution. And they can enter 3 different N's, and then they can click Draw New Sample at any time to just generate a new sample. So if N is 100, I think we can all be pretty confident that this looks normal, this looks skewed, and this looks bimodal. And most of us would have no problem there. When we get down to an N of 20, life gets a little bit more complicated. So this we could say looks probably normal-ish. This may or may not look skewed, might also look slightly normal. Hard to say. This does look rather bimodal, but we're starting to see some uncertainty here in how confident we can be. When we get down to N of 5, there's very little information, and they all look the same. Nobody has any idea what's going on. We can't tell. So we'll ask our students, okay, what happens if you have an N of 100? What happens if you have an N of 5? And then if you had to pick a number saying, where am I confident that I can tell, what number would I pick? And they can play with all kinds of different numbers and figure out what answer they would get. We then come and say, okay, what was visually with your eye? What happens if you do a normality test? How does the normality test work? So we have their same 3 distributions, normal, skewed, and bimodal. And first off, we need to talk about what we're going to expect. So if the data are coming from a distribution that's actually normal, we're going to expect that 5% of samples are going to fail that normality test, right? This is our type 1. 5% are going to be extreme in some way, so even though they came from a normal distribution, falsely identify them as being not normally distributed. On the other hand, if our sample came from a skewed or a bimodal distribution, it's definitely not normal. So we would expect 100% of those samples to fail a normality test. So what actually happened? Well, here we have the same setup, so we can enter our sample size. So we have 100, 20, and 5. And if we have 100, then it looks about like it should. We get 6% with the normal distribution and 100% with a skewed distribution. So just like our eye, the normality test is working well. When we go down to an N of 20, our normal distribution, we're still getting about a 5% failure rate, but we start to see something very different happening here for the skewed and bimodal distribution. Now, in this case, more than around half of the samples that are drawn from those distributions are still failing our normality, or are still passing our normality test. So the test is having a hard time distinguishing, just like we did by eye, as to whether or not data came from a normal distribution. When we're down to an N of 5, we have a 10% and a 12% failure rate instead of a 100% failure rate. So just like our eye has no idea, also the normality test has no idea. And again, students can enter any numbers that they want in here and get a sense of what do they get for additional 5 samples. Yeah? I'm not sure, but we can also change it and be, like, this is all done, so it can be changed to be whatever normality test we want to use. Yeah. Oh, the question was what kind of normality test was used. Okay, so in terms of solutions, I'll talk about a couple of things for static graphics and alternatives to Barnline graphs, but I regularly send things out from my Twitter feed as well. That's another place you can look for more information, and I'll probably send out a bunch of stuff tomorrow following this talk. Okay, so if we're not using bar graphs, what are we using instead, and how do we decide? Because there are multiple different options you should use. Well, it depends. It depends on your sample size, it depends a little bit on your data distribution, and it depends on the goal of your graph. So assuming that we have a continuous outcome variable and a small sample size, so less than my arbitrary number is going to be 15. It's an arbitrary number, telling you that, full disclosure. So in this case, you are going to use a dot plot. Why? As we've just seen with the simulator, summary statistics are only meaningful when you have enough data to summarize. When your data sets are really small, those summary statistics can be accurate. So it's best to simply show, this is my actual data, these are the points, this is the observations that I had. If you have a slightly larger data set, you now are going to get into the point where the summary statistics shown in a box plot are going to have some meaning, and your sample is large enough to actually calculate these. So in this case, you might consider using a box plot with the data points overlaid to allow readers to see not only your sample size and a little bit more information about the data distribution, but again, to still be able to critically evaluate the data. If you have a large data set, then it doesn't make sense to show the dots. You'll end up with a swarm of dots and it'll be really hard to see anything. So the summary statistics for your box plot are going to be accurate, and you can simply present a box plot. Critically important thing here, if your data distribution appears bimodal, you should not use a box plot ever. A box plot will mask a bimodal distribution, so there will be no way to see that in fact you have a peak here and a peak here. That information is going to be completely lost in the graph. So bimodal distribution, never use a box plot. The other alternative, if you're not a fan of box plots, I know there are lots of basic scientists who aren't, is to go with something called a violin plot. And you can show the data points on this, or you can take the data points off, depending again on your sample size. So again, useful for continuous variables, medium to large sample size, and you can use this with any distribution. So it will show a bimodal distribution if you use this type of plot. And essentially what you're doing here is you're taking all the data points the distribution. And the last thing is your bar graph. Your bar graph you don't use for continuous data, you're going to use it for counts and proportions. And it can be used for any sample size. So a lot of investigators have started to create box plots. When we're creating effective box plots, it's really important that all of the data points are visible. So I'm going to walk you through a series of strategies that you can use to make sure that all of your data points can be easily seen. So we start off with our ineffective graph here. We have a lot of data points overlapping. It's a little bit hard to know how the data are distributed and what's going on. So the first thing that we can do is decrease the size of the points. This helps, it cleans things up a little bit, but we still have kind of a mess with a lot of overlap. So the next thing we can do is make those data points semi-transparent. When we do that, regions with overlapping points are going to come up darker than regions without overlapping. The next thing we do is random jitter. So we add some noise on the x-axis, and again, this is designed to reduce the overlap. The problem with random jitter is it never fully eliminates the overlap unless you have a really small sample, and it also isn't very effective for showing you the data distribution. Or a more visually appealing option and visually easy to interpret is symmetric jitter, where you try to line points up on either side of the x-axis, or along the x-axis, so that they're symmetrically distributed and you can get a sense for what the data distribution would be. The next question that I'm trying to make these dot plot things, but I have a lot of groups in my graph, and my figure should give a clear indication of what my differences are, and nobody can tell it with these dot plot things, so I like the bar graphs better. So the first thing you're going to do is increase the width of your plot. White space. The next thing you're going to do is emphasize your summary statistics. So here I've shown my means and my error bars in black. So groups look different from other groups. But then I can also go back and look at the data points, which are in the background in gray. Sense for, does everything look like it's distributed appropriately for the summary statistics in the tests that are used? I can evaluate the data set in more detail. This is how you work with figures that are larger, that are more complex, that have a lot of different groups. So while our initial paper has been very effective in encouraging some journals to change policies, we've now moved to a different strategy for encouraging investigators to provide more information about their data and their data distribution. So we published a third paper in PLOS Biology called From Static to Interactive, Transforming Data Visualization to Improve Transparency. And the objective for this line of research is to really create the tools that investigators need to transform their scientific publications from static reports into interactive data sets narrated by the authors. So if we think about the incoming generation of scientists, and many of you are in the room are young, I'm sure you've all grown up with smartphones and tablets and every interactive app you could ever want, and the ability to manipulate a lot of data very quickly. This is the future of science. If the weather is interactive, certainly science can do better. And so we think that it's unlikely that as more and more of us get used to using these technology and tools, we're going to continue to believe that the best way to present our data is in a little tiny bar graph printed in black and white on a piece of paper. So we started off with the line graph. We had a lot of people emailing us and say, I really like being able to see the individual level data. A dot plot, that adds a lot of information, but how do I apply those same principles to a line graph? And there are a couple of problems with the line graphs. There are two pieces of information that we really want to have as scientists, and they're hard to get out of a line graph. The first thing we want to know is how much do different groups overlap? And depending on the line graph, this can be really difficult to tell. We often end up with something that looks like this, where we have a lot of overlapping error bars, and it's hard to tell which set of bars goes with which group the overlap is. The second thing we want to know is, do all the individuals in a particular group follow the same response pattern? There is no information about that on a line graph. A line graph tells us what happens to the group on average, but there are no individuals in this graph. So we get no information there. So to address this, we created a free web-based tool. Anyone can access it by going to this website. And it allows investigators with no programming expertise to quickly create an interactive line graph for their next scientific paper. So if you can, or check your email or check the weather online, you can do this. You can use this to make an interactive line graph. I'm not going to demonstrate it today, but there is a set of video instructions on the site that's linked that you can just go watch a video of me showing how to use the different parts of the tool. And I am going to show you a bunch of screenshots just to illustrate how easy this is and what kinds of things it can do. So the interactive line graph allows you to do 4 things. First, we can examine different summary statistics. Second, we can display lines for some or all individuals in each group. Third, we can view a subset of conditions or time points. And fourth, we can view change scores for any conditions or time points. First step is getting your data into the graph, and there are 2 options for how to do this. If you are a manual data entry person, you can enter your data manually. So you can tell it the number of groups and conditions, give it some names for your groups and your conditions and some sample sizes. And then you can either click on Save and Proceed to enter data, and you can type in your values, or you can download a template CSV file and download a CSV that you can open in Excel. It'll give you all of the headings and all of the forms, and you just type in your numbers into that. And you can then upload your work set into the data file. So an XML is for a graph that we've a CSV file is if you are creating a new graph with a new set of data, then you'd upload a CSV. You just select your data file, upload it, and the program will automatically create the graph for you. So when your graph comes up, you will see something that looks like this. And the first thing we've done is we replaced the error bars with a semi-transparent shaded region. So this is going to make it much easier to see which groups overlap. This is the normal mode. It has a colorblind mode, so it's just one click. If you're colorblind, click that, and that's much more easy for you to interpret. The next thing, the first thing you can do with the tool is select any option for your summary statistics. So you can show means or medians, you can show means with standard deviations, standard errors, or 95% confidence intervals, and you can also show a median with an interquartile range or a range. So that allows you to pretty quickly click through and see a lot of different summary statistics quickly. The next thing you can do is focus on specific groups, time points, or conditions of interest. So if I go back to my main graph, it looks like there's a lot of variability in group 2, the red group, compared to the other group. And I might want to know why that is. So one of the ways I could get that information would be to focus on group 2. So if I unclick the boxes next to any group or condition, it will remove those from the graph. And I can just add them by rechecking those boxes at any time. So here I've unchecked group 1 and group 3. So now I can just focus on group 2. The next thing I can do is view lines for any set of individuals in the data set. So I can select all or none, and this will show me the lines for everyone. And when I see that, it looks like I have some people that are responding and some people whose values aren't changing here in condition 2. And I can fiddle with this in more detail to figure out which individuals are responding and which aren't, and to just be able to look at the responders. The last thing I can do in order to confirm is I can go to the difference plot tab, and this allows me to create a difference plot for any set of time points or conditions. So I can change whether I'm looking at the mean or the median for the line here. I can select condition 1 to 2, but I could also do 2 to 3 or condition 1 to 3, any 2 conditions that I want. And then I can also remove groups from the graph if I want to focus on certain groups. And when I look at the data here, it does look like I have some non-responders and some responders here in group 2, which I don't see in the other groups. The Interactive tool also has some features for linking to publications. And in particular, we want to make sure that the static graphs that we present in our paper align with the interactive graph that we are giving people to play with. So there are some functions that allow you to save any static graph that you create using the tool in your paper. So I can simply enter a name for the figure. So here I have figure 1D for my paper. I click Save. Once I do that, the figure 1D is going to show up in the build menu, and I can put as many figures in here as I want. And this is a useful tool because it allows you or your reader to explore your entire data set, or just to say, I'm interested in figure 1D, I want more information on that, so I'm just going to go directly to that and start my exploration from that figure. And then the next thing you can do is download. So there's several different ways of downloading, and there are clear and easy options on the file. I can download a CSV data file if I just want to get the data out and run some stats. I can download TIFF files for any of the saved static graphs, and this is what's going to go into the printed version of your paper. And then I can also download an XML file containing your interactive graphic to include in a supplement for the paper. And that allows data graphs in your paper. Upload the XML file back into the site to explore the data set in more detail if they'd like to do that. Important thing to know when using this site, we do not save. I don't care. So it's important to save your work and save your XML file if you're going to want to go back and make changes to that graph later, because we do not save it. More recently, we released a tool called the interactive dot plot. So this is an alternative to the bar graph. It came out at the end of December. For those of you who are wondering about the title of the paper, we submitted it with a much more mundane title, and the editors asked if we could come up with something that would be more fun and a play on words, so we obliged. So now I have a scientific publication that includes the word naked in the title. All right, so the interactive dot plot tool will allow you to make a whole range of different types of graphs, and which one you're going to want to choose depends on your sample size and the other stuff we talked about. So you can look at a dot plot, you can add some error bars to that dot plot, or a box plot to that dot plot, and there are options for putting the dot plot in the front and in the back, or vice versa. You can also do violin plots, and this is because a lot of commercial software packages don't to make violin plots, so here is a convenient, free, and easy way of doing it for those of you who want to use those. You can have a dot plot with data points on it, or a violin plot with data points on it, or with a box plot in the middle. And we will actually even allow you to make bar graphs. But if you make a bar graph without data points, you'll get a message that will say, the bar graph is included as a teaching tool and is not intended for use in scientific publications. See relevant publications and initiatives. And those numbers will take you to links that will explain why it's a terrible idea for you to use a bar graph for your paper. You can do it if you want to. A tool has a couple of other features. You can examine subgroups. So for example, if you're working with a data set that includes males and females, you can actually allow your viewer to look at the data for the males and females separately. So you can color code the males and females, you can choose to show them side by side, or you can just show them all pooled together in one group. And you can add summary lines to show the median or the mean for your males and females. And I could also uncheck these to show only the females or only the males if I wanted to do that. The next feature it allows is for you to show clusters of non-independent data. So this is your replicates or your mice from the same litter. And it will automatically do a between group cluster design. So this is where I have one observation from each cluster in each group. It will do a within group cluster design. So this is where all of my observations from cluster 1 and cluster 2 are in group 1, and all of my observations from clusters 3 and 4 are in group 2. And it will allow you to do a within group cluster design. So I have observations from each cluster in each group. Okay, so when we're thinking about our interactive tools, we envision two sets of solutions. One is the author level solution. So these types of tools that I've shown you that allow you as to go in and quickly create an interactive graph for your paper. The second one is journal level solutions. So we're very interested in working with online graphics, a part of the scientific publication. So load some data and then upload it into our site. You can just play with it directly on the journal site as your online version of that paper. And we'd also like to make sure that those solutions are compatible. So the stuff that you create using our author level solutions, we hope at some point will just be able to automatically be fed into the journal website so that the journal can immediately use that, and you won't have to create another whole set of stuff when you're publishing, because we all hate that. Okay, so I just want to emphasize that changing data presentation for continuous variables in small sample size studies is really critical to promote transparency. So right now we have the bar and line graphs, and then we have everything else. And we would like to switch this around. So we would like this to be figures that show the data distribution, that are clear and transparent, and this is the bar and line graphs. And I would appreciate the help of everyone in this room in continuing to move that process forward. So here are some things you can do to help. The first and obvious thing is to banish bar graphs from your papers and talks. And if you are a reviewer or an editor for a journal, then I would encourage you to request figures that show the data distribution as a standard part of all of your reviews. It's also important to talk to editors for journals that you read regularly about the importance of improving data presentation in their journals and offering more transparent options. And lastly, I think it's important for investigators at any level to be working with statistics and presentation and statistics training, and things that would be appropriate not just for trainees, but also for junior investigators as well as senior researchers. And lastly, I want to emphasize that it's important to be a part of the solution. So I did not plan to have a partial career in meta-research. That was something that happened accidentally because I was frustrated by something that was going on that I felt was limiting my field's ability to move forward. And I'm sure that many of you in this room have similar concerns about other practices, be they in stats or in methods or any other area of your choice. So it's always a possibility to read meta-research studies, to learn about existing problems, discover better practices, identify problems in your field, and see how fields that may be further along in this area have problems, and whether there are solutions to your own field and into your own work. And finally, for those of you who have particular issues that you're passionate about and concerned about, I would encourage you to start talking to people and to bring together teams and perform your own meta-research studies to solve a problem that's of importance to you in your field. All right. I would like to thank my collaborators, so my mentor, Weissgerber at Mayo. The two statisticians I work with are Stacey Winham and Natasha Milik, who's the head of biostats at the University of Belgrade Medical School. Ethan does all of our simulators, and Marco Pro does all the programming for our interactive tools. And that I'm ready to take lab bursts. Okay, so the question is, how do we start addressing these concepts of statistics education earlier before students get to grad school, and how do we present them in a way that resonates with students? We don't have an undergraduate program at Mayo, so there's not a whole lot we can do there. So we are essentially dealing with graduate early career investigators who are looking for information, and sometimes PIs. So we do a lot of, we'll do our particular lab wants a seminar on how to present data or an instructional session at lab meeting. We will do a lot of outreach and things that way to get information to lab groups that are interested. I think what you said about targeting examples and setting up things in a way that's appropriate for the audience and will resonate with them is critically important. And that is the centerpiece of everything that we do. So every time we work with a new statistician, I give them a full one-page, 10-point font, no spaces list of everything that we know about our audience. So we talk about what study designs they're using, their level of statistics education, what software they're using, what mistakes are common in the field, what sample sizes do they work with. And then we have a full conversation about how the particular problem that we're interested in is going to manifest in the published literature for people in that field. So we make sure that our examples are just either one of two things, either discipline appropriate wherever possible, or general enough that people could see their own data in what we present. So things like some of the figures that I showed, they're very vague, but I think many of you in the room who are basic scientists could look at it and say, okay, yeah, I've seen data that looks like this. I get data that looks like this. So whenever we are writing a paper or doing anything, we spend, other than data collection, for our meta research, we spend about 30% of the time on content and 70% is on our communication plan. And how are we going to present this in a way that resonates with our basic science audience? So I would say, if you're not already working with a basic scientist, or two or three, find one, or two or three, because those are the people that can really help you in terms of saying, you know, because you're, as a statistician, you have a different mindset, you have a different way of thinking. So the argument that's going to work with a room full of statisticians is going to be the argument that's going to work with a room full of basic scientists. So having a basic scientist that you collaborate with closely, I think, is really essential in making sure you're getting the right information to the right people and at the right time. Many things are possible. Seeing the data is really important. And it's important for a lot of reasons. So the question was about papers that have bar graphs with a lot of groups that overlap, and the error bars also overlap, but the differences are significant. So I think one of the things that's critical and that's a problem with basic sciences is a lot of basic scientists aren't aware of what the error bars represent. So really checking, is it standard deviation versus is it standard error? Because with standard deviation, you will get a lot more overlap, even if there is significant difference. So that would be the first thing I would look at. And then I think it's also really important to think about the statistics. Was the technique that they're using appropriate for the data that they were analyzing? And so we just finished a study on reporting for ANOVA where we were asking the question, among papers published in top physiology journals that were using ANOVA, can we tell what type of ANOVA that they did? Do we have enough information to verify the test result? And we also looked at the prevalence of 2 common errors. So papers using a one-way ANOVA when 2 or more factors were needed, and papers that were not using repeated measures or didn't say they used repeated measures when there was a variable that clearly seemed to require repeated measures. And the results were horrifying. And it's surprisingly frequent that the stat section is just a statement of data were analyzed by ANOVA as appropriate, significance determined at p less than 0.05. In that case, you have no idea, and what is as appropriate, especially when your stats education isn't standardized. That can be mean very different things to very different people. So one of the things that we're pushing towards now is really encouraging investigators to report. So I can tell what stats were used. I can tell I have the information there to verify the test result. And if there is a problem with investigators using a statistical test that isn't relevant to their data, I can pick up those problems. Well, okay, so the question is whether there's any sense of how prevalent the same problems are in social sciences, which also have small sample sizes. I think there's a very active research group in psychology that's looking at these issues and has found many of the same types of variables. And in fact, for things like bar graphs, there are papers showing that practice is common in psychology as well, which we cited when we did our original paper. So I've seen a lot there. I've seen less on the other social sciences, but that may just be because there isn't the same level of interest right now. Psychology knows that they have a problem with reproducing findings, and they're really committed to, they have a core group of people that's really committed to solving that problem. So that's where I've seen the most. Doesn't mean it's not happening elsewhere. It's just based on what I read, that's what I see. Okay, I talk to editors on a regular basis. One of the things, so the question is any suggestions for editors about how to approach your editorial board about getting changes made. When I talk to editors, the ones that are able to make changes are often saying, we have a lot of people who like this. We have some people who are dead set against it. We happen to have people in the right positions who like this, and that's allowing us to move the changes forward. So I think that is a critical piece, and you know best what the makeup of your board is. I think presenting information and papers that are really written for basic scientists are important. So ours is, there's a couple of others that I can give you that are as well. I think emphasizing what other journals are doing. So I have a list of journals that have made policy changes with regards to their statistics, that have added checklists, that are doing things on data visualization. And so checking other journal policies and how specifically they're wording things and what they're doing can also be something that encourages journal editorial board members to make similar changes. So those are all strategies that I would recommend. The other thing is, if you can get people to make changes in their own lab, they're much more likely to be comfortable with encouraging others to make those changes. So we found that if we can get people to start graphing their data in dot plots, it fairly quickly becomes obvious to them why bar graphs are such a problem. And we don't have to do a lot of other explaining because they just get it. So anything you can do to encourage journal editors to try things out in their own lab before they push a policy forward, I think can also be a help for convincing. From what I see in physiology, we're so far away from having a problem that I'm not too concerned about it. That would be my answer. I mean, I think with the type of literature that I deal with, we are thinking about, you know, how can we get people go from 5 to 10 or 15 or 20, which is still within the point where there's a lot of inaccuracy in those summary statistics where there's low power unless you're looking for a really big difference. We're not at the point where we're, you know, thinking about 500 versus 1,000, which is where some of those other detecting really small differences that aren't clinically meaningful become an issue. Yes, it will definitely have an impact, and so we tend to think of power as being sample size. Power is not just sample size, it's also the amount of variability in your data. So if you reduce the variability, then you increase your power, and it's also the difference between your groups. So the bigger your difference between means or median is, the higher your power. So when you're thinking about increasing power for a small study, it's not just increasing sample size, and if you can't do that, there's nothing you can do. There are other things, and that may be one of those areas where if you have large differences as well, you do have relatively high-powered studies just because your variability is so low. So I would say Daniel Lakens, so the question is about, is there a statistics course online that I would recommend? Daniel Lakens has a good course through Coursera, and I don't remember what it's called offhand, but I think it goes, and it's geared a little bit more towards a psychology audience, but it is general enough that it's still very useful for basic sciences. So I think that has some very good, there's a lot of simulations and things to help you understand, like P-curves, how do those work, how are they affected by power and sample size, and those types of the start. And then I think Twitter is also a really good resource for getting information on all things meta-research. So I would look there and start to find and follow meta-researchers on Twitter, and they're going to be sending out papers and resources and links all the time that can be very helpful to you.